My title is A Natural Approach to Pancreatic Cancer and Pancreatic Cancer Recurrence. I want to say at the outset that I'm talking about pancreatic cancer, but the natural approach I'm referring to, which consists of two anti-cancer plant extracts, this approach is effective against other kinds of cancer and other cancer recurrence. So there's a very broad spectrum effect of the natural approach underlying the specific story I'm going to tell you today. Early pancreatic cancers often do not cause any signs or symptoms. By the time they do cause symptoms, they have often grown very large or have already spread outside the pancreas. So it makes them very difficult to treat because they're diagnosed late and they're advanced. And in fact, many of the symptoms that are associated with pancreatic cancer are more likely to be caused by other conditions. So for example, lower back pain, diabetes is another case. But by far and away, most of the time, those two conditions are those conditions. A diabetic has diabetes, not likely because of pancreatic cancer. So in a sense, the disease is hiding behind its own symptoms, and it gives cause to what Dr. Bard has referred to as pancreatic cancer as a silent cancer. So let's look at the DNA in these cancer cells and consider Beljanski's theory of cancer. You see at the top here, just a diagram of the DNA duplex. It's two long strands of DNA which are spiraled around each other, and they're stabilized by hydrogen bonds, weak chemical bonds that are depicted in the little vertical bars between holding the two strands together. So those hydrogen bonds stabilize the DNA duplex. Beljanski reasoned that the DNA structure of a cancer cell differs from normal DNA structure in its secondary, not just its primary structure. So he wasn't focused on the DNA sequence, he was talking about the physical, chemical structure of the DNA. And in fact, how stable the two strands were, how stable the duplex is. So he made the prediction that, that cancer DNA was structurally different from healthy cell DNA because of this chemically damaged structure. The DNA, the hydrogen bonds are broken, and as a result, the structure is opened up or destabilized. Well, let's look closely at how this happens. Oxidative damage and carcinogen binding lead to DNA destabilization. So at the top, there's a healthy, tight DNA duplex. Oxidative stress is a major target. One of the major targets of oxidative stress is the DNA in the cell nucleus. It is chemically damaged by this, and this damage, which includes stretching of bonds, substitution of atoms, and deletion of atoms, can give rise to the earliest stages of destabilization. Little loops can form because locally, the base pairing, the hydrogen bonds are weakened and broken, and there's a little pocket or loop that is present instead of a stable duplex. In the end, what he realized was when you add carcinogen binding, which further destabilizes the structure, you have separated strands. And the enzymes for DNA replication have increased access to the duplication sites located inside the double helix, and duplication can become abnormally accelerated. So this abnormal acceleration of DNA synthesis is an absolute feature of cancers. I want to point out quickly that cancer DNA is destabilized DNA, and Beljanski's observations have been vindicated. His work was reproduced in a completely separate laboratory in Seattle, Washington, by a man named Don Malins. And just look at the title of that first paper that Malins produced here. Infrared spectral models demonstrate that exposure to environmental chemicals leads to new forms of DNA. He's talking about a new structure, and he's in fact referring to DNA destabilization. He's not referring to DNA sequence. It's very important that that distinction is clear. So what did Beljanski do with this insight? He had a prediction that DNA synthesis would be accelerated in cancer cells, and he realized that that's the basis of a test. It's testable. So he called it the Oncotest, and he could include in test tubes the DNA template from a healthy cell, purified DNA, and compare that to the DNA from a cancer cell, again, purified DNA, the other ingredients that are necessary for DNA synthesis, and then see what happens. And in fact, there's a slight difference. As he predicted, the DNA in the cancer cell template is generating a little bit more DNA synthesis than from the normal cell template. But in the Oncotest, he realized he could take a step further and find out what the effect of any compound was on this synthetic reaction. And what he found was that known carcinogens provide a dramatic acceleration of the DNA synthesis in the reactions when we're talking about the cancer DNA template. The normal DNA template is not much affected by the addition of these exogenous carcinogens, but the cancer DNA template is extremely susceptible. And you can see that several examples. DMBA is a really horrible carcinogen. Vinblastine is a chemotherapy drug, also a carcinogen. And steroid hormones have this cancer effect when they're used at high concentrations. They have a destabilizing effect on the DNA, on the cancer DNA. Some compounds, as you can see in row B, don't have any effect. They don't interact with DNA. Some in C, diquat and paraquat, poison the reaction by interfering with the enzyme polymerase that makes the synthesis. But in D, we have something really interesting. What would be the behavior of an anti-cancer substance? It would shut down DNA synthesis from the cancer DNA template while leaving synthesis from the normal template alone. Beljanski screened for such substances and found two, Pauperera and Rauwolfia vomitoria. The Pauperera is a tree that grows in the Amazon rainforest. Rauwolfia vomitoria is a shrub that grows more widely, but it certainly is in Western Africa. The most active compounds in these extracts were identified by Beljanski, and he went on to show that they do indeed have anti-cancer effects against cells. Predicted in the Oncotest, they also work against cancer cells. And there's a building here from the Pasteur Institute where Beljanski worked for many years. And then 3 experiments showing the effect of Pao and Rauwolfia extracts against 3 different human cancer cell lines: human liver, human breast, and human thyroid. And those vertical bars are showing the results. As the concentration of the extracts are increased, the number of cells, cancer cells, decreases. And blue is Pao, green is Rauwolfia. They both work in similar extent to kill the cancer cells, to reduce the cancer cell population. In one of his later papers, he reported on what he called PB100, another name for Pauperera extract. He showed it's selectively active on malignant cells, inhibits multiplication of 16 malignant cell lines, even those that are multi-drug resistant. And among these cell lines were included a pancreatic cancer cell line. So just to pause and point out that what Beljanski did was produce a model of carcinogenesis and also show that the DNA destabilization he saw as fundamental to carcinogenesis was also an attribute of virtually all cancers. This damaged DNA structure, this destabilized DNA, is found whenever you have a cancer cell. If you look at the DNA, it has this defective structure. So it's broad spectrum. DNA is DNA. All our cells have it. If it's a cancer DNA, it's destabilized. And that means that the extracts will recognize that and actually end up killing those cells, the defective cancer cells. It's also, I'm not going to talk about it today, these extracts are effective at early, middle, and late stages of the disease. So precancer, mid-stage cancer, and advanced cancers. And as I already pointed out, he included a pancreatic cancer cell line in this panel. Well, let's look at some newer experiments from Kansas University Medical Center, showing that Pao and Rauwolfia selectively kill pancreatic and ovarian cancer cells. I included the ovarian cells here because these were done in the same time frame, these experiments, and ovarian is another late diagnosed, very difficult to treat cancer. And we were very interested in seeing how the plant extracts would do against these very difficult to treat cancers. And this is just simply a cell-based study, similar to what I showed you that Beljanski did with the 3 cancers. Here, pancreatic cancers, there are 5 different cell lines. The Rauwolfia and Pao extracts kill these cancer cells, and they do it specifically or selectively. The top line in all these graphs is MRC5, which is a normal cell line. It's not a cancer. And you can see that the extracts are not active against that compared to their activity against the cancer cell lines. So this is an animal experiment which looks at the effect of the extracts on tumors in animals, pancreatic cancers. We again find that these extracts do their job by inducing apoptosis. And just to look briefly at the Powell-Pereira treatment on the upper right, the control is untreated. That has the tumor grows unimpeded. Gemcitabine is next, which shows some reduction in tumor growth. And then the Powell, interestingly enough, Powell is better than gemcitabine alone. And that's in part because these cancer cells become resistant to the drug. Gemcitabine is often the drug of choice or first line choice for treatment of pancreatic cancers in humans. But key point here, it's the combination of gemcitabine and Powell-Pereira that does the best job in shrinking these tumors. It's very significant shrinkage of the tumors. The other thing that this combination does, and Powell does alone, is reduce the number of metastatic foci. So we're impeding not just growth of the tumor, but also metastasis. Here's a summary of these kinds of experiments, just showing graphically that the control in blue is unimpeded growth, untreated. Gemcitabine is next, and you can see it has some effect, but nowhere near as good as the plant extracts by themselves, and nowhere near as good as the combination of Powell plus gemcitabine. So to summarize Powell treatment, at the end of the experiments, the internal organs were tested and histologically analyzed. No tissue damage was detected in any of the groups. The Powell-Pereira extract exhibited strong inhibition of PANC1 tumors, that's a pancreatic tumor type, throughout the course of the experiment, reaching 70% inhibition, even when the tumors did not respond to gemcitabine anymore. And this phenomenon of using the two together, gemcitabine, the drug, plus Powell-Pereira, has a better effect than gemcitabine alone, and also slightly better than Powell alone, but it also enables what's called a dose reduction effect, which means that the toxic chemo drug can be used at a somewhat lower concentration and get the same cytotoxicity against the cancers while alleviating the negative side effects of the drug. Similar positive results obtained for Rauwolfia against pancreatic cancer. And again, these results refer to potentiation of the gemcitabine effect when the Powell is included in the treatment, or the Rauwolfia is included in the treatment. In a sense, the extracts have the effect of reversing the drug resistance of the tumors. So this is another important take home here. Synergy with multiple chemotherapy drugs. We've looked at lomustine, docetaxel, and carboplatin, which all work by different mechanisms, and they all are synergistic with these two plant extracts. So I think there's a big future here for more effective cancer therapies when these extracts are included. So let's move on to cancer recurrence and ask the question, what are stem cells? The stem cells are capable of perpetual cell division and self-renewal. And there are two types of healthy stem cells, embryonic and adult. And you can see on the left here, this panel, the ultimate stem cell is the fertilized egg. It's a single cell fusion of sperm and egg, which then gives rise to an entire human being or animal, whatever species you're talking about. So embryonic stem cells enable growth and differentiation of the body, whereas adult stem cells, we have them too, they enable renewal and repair of tissues. So both embryonic and adult cells have therapeutic applications, but that's not what I'm talking about right now. There is a third type of stem cell, cancer stem cells. These are a small subset of the cells in a tumor that have hijacked the normal stem cell potential to divide and persist, enabling survival, resistance to chemo drugs, and metastasis and new tumor growth. So cancer stem cells are what enable cancer recurrence. Brief characteristics, cancer stem cells are a minority of the population, but they survive after drug treatment. and persist. And sometime later, sometimes many years later, they are the basis for tumor regrowth, or what's called here relapse, the same thing as recurrence. So strategies to eradicate cancer stem cells are an urgent topic in cancer research. Well, let's see how the two extracts do against these cancer stem cells. The first approximation are in vitro. It's called spheroids, and that's a phenomenon that stem cells do. They attract a cluster of cells around them. So the other cancer cells glom on to the cancer stem cell, and you get these, as you can see in the control in the upper left, there's big spheroid, it's called, a big blob of cancer cells surrounding a cancer stem cell. And in both experiments, Pao and Rauwolfia destroy these spheroids. So this is the first indication that there could be activity against cancer stem cells. What's important? How do we define a cancer stem cell? How do we know if something is a cancer stem cell? It's positive for 3 cell surface markers in the case of pancreatic cancer stem cells. These markers, in a sense, are identifying the cell as a stem cell. And these markers are shared by, for example, embryonic healthy stem cells. So there's a real recruitment of the stem cell identity here, which is shown by these cell surface markers and the conferral of stem cell division and propagation effects in a cancer cell now. It's being harnessed and stolen by the cancer cell. These cell surface markers are great because they enable the experimenter to isolate, to purify a population of pancreatic cancer stem cells. And we can then see what happens when the 2 extracts are administered in these cultures. And in both cases, both Pao and Rauwolfia, the cancer stem cells are killed off to varying extents. It's more highly specific, efficient in the Pao than the Rauwolfia, but both of the extracts work against these triple positive pancreatic cancer stem cells. Well, let's look then at the key experiment here, the effect of the extracts on tumor formation by pancreatic cancer stem cells. And these experiments, those stem cells I just told you about, purified pancreatic cancer stem cells, are injected into the pancreas of the experimental animal. And the tumors are then allowed to grow, and they're either treated or not. The control is no treatment, unimpeded growth. Pao and Rauwolfia are the 2 extracts that are used here. And you can see treatment runs through 24 days, and there's a significant decrease in tumor formation both by Pao and Rauwolfia. And it's interesting to note, 100% of the untreated animals get tumors. There's no stopping these pancreatic cancer stem cells. But both extracts significantly reduce the population of animals that get these tumors. And this is a wonderful result. And I can show you then, we can have an effect. The extracts can limit the formation of tumors, but also limit the growth of the tumors that do form. And here again, the red line is untreated. Those tumors are taken off. Both Rauwolfia and Pao Pereira inhibit the growth of the tumors that are initiated by the pancreatic cancer stem cells. And it's important to note, so we already get down to 60%, so there's no tumors forming in 40% of the animals that are treated with Pao. But I want to emphasize that the tumors that do grow in the other 60% are smaller and there are fewer foci. So it's overall a dramatic effect of the extracts against the pancreatic cancer stem cells. Well, finally, I've told you about cells, I've told you about animals. How do these extracts do in people in the case of pancreatic cancers? And there's one testimonial I have to conclude here from a nameless individual who you'll kind of get to know when I read it. Since the beginning of December, I have ordered Pao Pereira and Rauwolfia vomitoria for my son who has pancreatic cancer. He had a 4-centimeter tumor on the head of his pancreas. After 2 months treatment, his tumor had already shrunk by 1/4 and is only 3 centimeters long. I wanted to thank you for the effectiveness of your medications. I couldn't talk to the doctor about your extracts because the doctor can only see through chemo. My son did not do well under chemo because he did not support the treatments very well, and this made him lose a lot of weight. I hereby confirm the effectiveness of your products. I have already recommended them to other people. So there's a testimonial to the effectiveness of the extracts in human beings, in a human being with pancreatic cancer. Thank you very much for your attention. I'm open to hear from any and all of you. My email address is there. And please, if you have, after this Q&A today, if you have any other questions, feel free to contact me.